Have you ever wondered how cells are able to reproduce? Well, there are two different ways cells replicate and reproduce: the mitosis and meiosis. Mitosis occurs both in haploid and diploid cells, but meiosis occurs only in diploid cells. Haploids are one pair of chromosomes, while diploids are two pairs of chromosomes. Mitosis results in the creation of two genetically identical daughter cells, while meiosis results in the creation of four genetically different daughter cells. Interphase 1 is the growth period where DNA replication occurs within uncondense chromatid. Centrioles and aster form, and a nucleolus is present. Most cells spend their growth and development during interphase.
In prophase 1, the nuclear envelope begins to break down, and chromatids condense into chromosomes. The spindle apparatus begins to form. Each chromosome pairs with its homologue, and crossing over occurs. Crossing over is when the DNA of non-sister chromatids are broken and rejoined to each other. Homologous chromosomes, or homologs, are a pair of chromosomes that carry the same genes from both sets of parents. The two homologous pairs combine into a structure called a tetrad. The crossing over is indicated by the color exchange of the purple and blue.
The next phase, prometaphase 1, in which the microtubules and poles attach to two kinetophores in the centromeres of the homologs, and the homologous pairs move towards the metaphase plate.
When the homologous pairs move towards the metaphase plate, they line up in an action called metaphase 1.
After metaphase 1, the homologs move towards the opposite poles, guided by a spindle apparatus, and the sister chromatids move as one unit towards the same pole because of interchromatid cohesion at their centromeres.
In telophase 1, two haploid cells form and each chromosome still consists of two sister chromatids. The spindle breaks down, allowing the cleavage furrow to form. Because we are dealing with an animal cell, a cleavage furrow forms between the two cells. Then the cytoplasm divides in an event not part of meiosis called cytokinesis, and the original cells create two haploid daughter cells.
Now meiosis 2 begins and no chromosome duplication occurs between meiosis 1 and meiosis 2. Within prophase 2, the nuclear envelope begins to break down and the chromatids condense into chromosomes. The spindle apparatus forms and the aster and centrioles become present.
In prometaphase 2, the chromosomes move towards the metaphase 2 plate. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical.
Next, the chromosomes in metaphase 2 wind up at the metaphase plate, and the kinetophores of sister chromatids are attached to microtubules extending from the opposite poles.
The chromatids begin to move towards the opposite poles as individual chromosomes during anaphase 2.
The nuclei form and chromosomes begin decondensing, while the nuclear envelope forms. The spindle apparatus breaks down and cytokinesis occurs. The division creates four daughter cells, split each with a haploid set of chromosomes that are genetically distinct from the parent cell and one another.